that's like the primo, but we didn't even see any yesterday.
So the bridge left for a minute, so does that mean we have to pause here?
Yeah, we're gonna have to pause for just a minute.
Okay.
5 meters.
So when you say, when are we looking for something that's like fist size or smaller than that?
Which size?
Fist, my
Oh,
hand.
fist.
Oh, I heard this.
I thought you said. I heard this too. This size or this size?
No, no,
Like, are you
no,
tons
that
again?
size.
So it'll be probably, if we found a 10 centimeter one, that would be awesome.
Okay.
So that would be big. So while we're sitting, we can
Oh.
just kind of zoom in and look at the rocks.
Yeah, we have a little bit of a ship move left, but we'll otherwise
Okay,
kind of hang out and
that's fine.
wait for the bridge to get back on.
That's what I thought I heard. Is that yeah, I see. We're still doing the move. Gotcha.
Tube sponge or barrel sponge looked like a desirable place to live. Such a neat little home.
So once we do the move, if we find some more of those tube sponges, Abby does have that on her list. So they've been intermittent in and out. So if we see some after the move, then we can grab some.
Sure thing.
So the sponges that are shaped like a vase, on the left-hand side, I see 3 of them. Are those still healthy or? Because they're
The
so
orange
porous
ones?
looking? Mm-hmm.
So is
Yeah,
this
that's
this tube sponge coming up into center of the lasers? You said this is
that
up
is
for collection?
definitely. I believe so. If we zoom in on, I can tell you.
Sure.
Just
You can just
10 meters. Or I'll just.
You can just hold off. Yeah. Go ahead, Marley. Push on in.
We also have lots of sea pens on the bottom now, the brownish, fluffy things. Yes, that is up for collection.
Up for collection, Roger. All right, go ahead and come wide, Marley. I think we'll be good because we're pretty flat right now.
Yeah.
And we don't have any, I think we got most of the swing out.
Mary, do you know what sample number this is?
This is sample number 065.
And you guys want a small piece, big piece, slurp, I would assume, for the small guy?
If you could get a tube base and the whole tube, maybe.
Tube base and whole tube, Roger.
Not like the whole organism, but just like one tube.
Sure. See what we get.
And if you look to the, what is that, the upper part of the screen near the middle, under the white sponge is a dead sponge with coral growing on it, the white fuzz. There's actually 2 different. Yeah, it's on the dead. Looks like a dead sponge underneath, and it's growing on it. Looks like clavularia.
Yeah, it might be takeable, the whole rock. Video, go ahead and push on in.
Yeah, it is pretty small.
Yeah, that'd be great.
Oh. Did you want this one, Kim? Reg. Good. It's all good. All right, go ahead and come wide there. All right, and so Mary, can you tell us what sample bins we have open on the starboard side?
We have E and F open, along with B, C, and D.
So we have our pick of the lottery here, don't we?
We really do.
I know this one was backwards, right? All right. We had some large. Six. All right, Marley, you want to push on in there a bit, please?
Yep.
That is great. Thank you.
Wow.
Open
Perfect.
K.
Little bouquet.
Yes.
Way easy. Way
For
easier than the
you.
coral yesterday.
All right, Marley, you want to push on in there a bit, please? And we'll get a screenshot of it. Get there. All right, go ahead and come on wide.
The Grenadier wants a screenshot.
Yeah.
Some cameras.
Cameras.
You took my flowers. I've been tending them so long. So did I looked at what Abby put together for sampling. Do these types of sponges have a taxonomic designation already, or we're just calling them the tube sponges?
They're unidentified tube sponges.
Okay.
Mary, remind me again.
How about we go with E?
So this could be the holotype of the whole thing. Or do you need more than one to do that?
Yeah, that it could be.
Sorry. Thanks, Kim.
Success.
Nice job, Jess.
Thanks, guys.
All right, before we leave, if we could take a zoom of that coral that's now kind of middle left underneath the white sponge, the white fuzzy stuff.
Middle left. Roger that. All right, Marley, go ahead and push on in.
Oh, it's even growing on the sponge.
Oh, sorry.
And the sponge is growing on a dead sponge.
Sorry.
It's not.
It's like the trees that grow out of dead trees.
Thank you.
Yep. That's good, thank you.
And all
Sure thing.
and all those little white stalks that are sticking up out of the mud, are those
Do you
worms?
want to continue on through?
Yes, they're either worms or amphipod tubes.
I can give you a sec if you need.
Yeah, sounds good.
And then right in front, can you turn the lasers back on?
Sure.
So right in front, we have a feather boa sea star. The reddish brown thing almost dead center.
So remind me of the heading that we're gonna want.
Yeah, just get up to this local max. We're gonna do 350 like that.
350. Roger.
Is this
Alright.
what you're talking about, Tom?
Yeah, right there. That's just a sea star that called the feather boa. Again, scientific naming or common naming.
All right, guys.
There's a
Inspect that
yep.
as
Would you
We
want
can
to
keep
stay here
going.
any longer? All right.
No.
350.
call it.
350, correct? Yeah. The rock was very helpful for that.
Yeah, it's all intact now, which is nice. It's going on a journey.
A journey it probably doesn't want to take.
No.
Just gonna reset you while you.
You can see all the feather boa sea pens all around the bottom. Just by moving a few meters, the whole bottom changes. And
Looks like we're on this kind of flat ditch.
Yeah, so yesterday when we were on the other slope, we didn't see any of these sea pens. It's interesting that here they are.
Yep, and earlier they were seeing different kinds of sea pens, not the feather boas. So they landed in 800, 700 meters, and so now we're up a few hundred meters, and we see a big difference in the mud community.
that orange rubble is sponge?
Yeah, I don't know what that. I was looking at that.
Maybe a sponge with some orange thing growing on it.
That could be.
It's like a
Do
lot
you
of
want
things
to check it
caught
out?
as a sponge.
Marley,
Sure.
you wanna do a quick zoom?
Yeah,
Looks like
sure
it's probably
thing.
just trash.
It looks like it could be trash.
No, it's like going over a
Or is it a-
junkyard.
Yeah. I'm thinking
I hope
that's
you're right
trash.
on it, at least.
Some folds.
Yeah. Well, with the sponges, with their different forms of decay, how they're crushed in on themselves.
Go and come by, Marley.
Yeah. It doesn't look like it's any kind of encrusting sponge or tunicate that's the orange, so it probably is some kind of trash. An old box. You can see that encrusting kind of coral on that old sponge right there.
That
Any
fuzzy
hard
white.
surface. Yep. That's exactly what you
Yeah.
look for, is that fuzzy white stuff. Or that look.
And I guess the sponges, because they have the framework of the spicules, and those are hard, they become a type of hard substrate as they die, with the spicules still remaining.
That is very true. And then you
Yeah.
also get, for these ones, these heterocones that have this kind of waffle pattern, it already has natural holes for things to move in and out of. So brittle stars definitely love being in there. You see them all the time. Some octopus coming up. Oh no, never mind. That's a more orangey looking thorny head.
Sometimes we just see what we want to see.
That's true.
Versus the blue thorny heads that are out there.
Yeah, there's a poacher going by near the bottom now. Something different.
Great.
There's a still upright dead sponge. It's interesting how there's just intermittent dead ones. You see a lot of live ones, and then you see a few dead ones, and then some live ones.
So with the number of dead sponges that we've been encountering, as you were saying, is it something like a virus is in the water, like the virus that swept through the sea star community, the nearshore sea star community? Are we thinking that it's something like that, or just, well, I guess we're just still trying to figure it out.
We are trying to figure it out, and
Yeah.
that definitely could be. There's a place
Mhm.
in the Catalina Basin that we've been looking at in 2011, maybe 2013. We went and sampled that with Liz Clark's AUV, and we looked at this community of sponges that was down at the very bottom of the basin, 1,200 meters, and it turned out that almost all of them were dead when 20 years earlier they were alive. And it was like a widespread death. And trying to figure out what caused that is one of the things we've been working on.
Yeah, like a pandemic.
And then we went, it was like that.
Yeah.
So it's a question of, did they lose oxygen in the basin?
Right.
And the only thing left is the sponge skeletons, because the fish would have been gone already. Was there a big sedimentation issue that covered all the sponges? Was there some kind of localized virus or something like that that killed them? We don't know. We took samples. I don't know if people have gotten anything out of it, because it's just the skeletons left. If you don't have a live part, it's hard to isolate something like that.
Right. Well, it's just, if it's a virus, it's hard to isolate to begin with.
Let's see if we can
That is
tell.
true.
Like
Yeah,
a
from
shofar.
ocean water. So
Here's your Rhabdicaulyptus barrel sponge, this brown one. It's still healthy. It's white inside.
looks like there's something inside of it too.
I think it's sediment.
Okay.
Marley, want to do a quick zoom? It's great there.
Seems like a liability to be able to collect so much sediment. Oh, so you're filtering.
Yep. Looks like there's some brittle stars maybe mixed in that sediment.
All right, go ahead and come wide, Marley.
Yeah. It's chunky sediment otherwise.
It is pretty chunky for being inside there. So if you look under the rocks sometimes, when we go up slope, you'll see a grayish bluish fish, which I have not seen this time, called a ruby-nosed brotulid. They're actually hard to survey and sample because they pretty much every time I've seen them, they live right under a rock. And the only time I really see them is doing this, going upslope where you can kind of see under the rocks.
Hmm.
There's an octopus. I got that one.
Oh yeah,
Oh,
good
yeah.
eye.
I've only watched a million hours of video and been doing this for 30 years, so you
Close your
start to
eyes
see
and
different
this one.
things. Yeah, though that is the sad part.
All right, Marley,
Tetris effect.
go ahead and push on in there a bit.
Wonder if it's gonna get mad.
Yep.
Just a
Good
little
morning,
flare.
buddy.
Yeah, getting a little agitated. Not sure what our intentions are.
All
Just siphoning
right, go ahead
water.
and come wide, Marley.
It's all right,
I'm
dude.
going to
We
put you on the
respect
cover
you.
of Octopus Weekly.
Good monthly.
Yeah, we don't get them long enough yearly.
So it looks like there's more yellow sponges.
Yeah, so these are Mycale or Mycale, M-Y-C-A-L-E. And they grow vertically like that in a sea fan, kind of like a coral would in a sea fan shape.
There are also just a lot fewer fish here.
Yep.
A little piece of kelp.
Yeah, I don't know how much sampling of the kelp, if they want one a day or how many they want it. I will have to look that up.
Oh, for the plastic.
And for
Yeah,
the microplastic.
it's for Taylor Ann, so I guess she's asleep.
Well
If she's
deserved.
smart.
She just got off shift.
Oh,
I think
Yeah,
it's
I
kind
don't
of
think-
opportunistically,
yes.
things like
If
that.
and if we had, yeah, I think you're right. And if we had sampling boxes to put it in. So if we saw some, I don't think that they collected any yet on this dive. So if we see any, we could just, knock it out.
Yep. Sounds good.
But we can continue along.
Yeah.
Okay, great.
Anything
Yeah, we
that
don't have to
see-
go back.
We don't have to go back. That's not what I meant.
Right.
You're right. Anything that we see moving forward on this shift.
But if we find gold doubloons, we're going to stop and sample them.
I see the motives now.
It's to pay for more ship time.
Yeah, I'm curious to hear how difficult it is to get like a cause of death for sponges.
It's pretty difficult.
Yeah.
I mean, some is easy when you see everything else is sedimented in around, but
Yeah, I used to think the sedimentation was the cause of death a lot of time when you'd see that, but I guess that happens after death too.
it can.
Yeah.
But it can be the cause too. Because they have to have enough flow to be able to stay alive. So if they're covered in sediment, they can't have water flow through them. There's a pair of
Yeah,
gorges.
so what's at the base of that?
That's a
Paragorgia?
Pharrea. That's a of the Paragorgia or the Heterocone. The Heterocone has a Pharrea sponge on it.
Okay, that's
So-
the one I made.
So if we want to zoom in on that one right below the lasers.
Sure thing. All right, Marley, go ahead and push on in.
It's also called the lace sponge.
It's pretty.
The Pharrea is the lace sponge.
Yes.
Oh, it's very pretty.
And the yellow one is Heterocone or Heterocone, depending on how you want to pronounce it. And it's also called a goblet sponge or a goiter sponge. And if you know what goiters are, you can know why it's a goiter
Yes.
sponge.
Something
And if
very
you actually-
medieval.
If you look to the lower right there, there's some baby ones of the Heterocone starting to grow
Oh, yeah.
on it. Just starting. 3 of them, it looks like. But you can see this look is different than the ones that have the big wide open top. This one is more enclosed. So is this a
Alright,
different
Marley,
species or a
mine?
different growth form? There's actually 5 different sponges in that picture.
It's pretty.
Then there's an aurora rockfish, all whited out. So how far have we gone since the last end of transect?
Yeah, let me check on that. Yeah, we've come 160 meters.
Are we set up well to do one? Does it look good up ahead?
Yeah, so we were going to go up onto this local max, which means we'd have to come back down.
All right.
We could instead continue along this contour for, let's see. I mean, we could do, yeah, we could either go over this feature and we have about 300 meters. It doesn't look like it's going to be much of a downslope, or we could go along the contour we're on around this maximum. So whichever.
Which way we're talking about going west around it?
Yeah, either kind of more westerly on this contour or kind of jump over and keep on it kind of a little more to the north.
Yeah, either one works for us. Which one would be better for you guys' sides?
It wouldn't matter. Whatever you feel would be easier to fly for
Roger.
you. Or if they're the same, I would say we just keep going straight then.
Going straight. Yeah, that works.
Yeah,
And these are 200 meter
Okay.
200 meter transects.
yeah.
Yes,
Yes.
let's shoot for 200 meters.
Sounds good. Yeah.
All right,
'cause
let's just keep 350.
So this is our 4th transect.
Yep, that's what I have.
Okay, me too. Just let us know when you're ready to start.
if it gets a little steep, then we'll maybe bump it down, but we can start with 40.
Yeah, just do what's best for you all on ROVs.
Roger that.
Yeah, it also works for us going down slope. You can't really see much. A lot of dark views. Here comes little baby Hake.
Yeah.
So I take it that's how much the current's flowing, how much that fish is moving. Well, now he's not. Now he's kicking forward. But
I guess if I take my hands off the sticks, you can kind of see the way the current flows.
yep.
So,
It's sort of pushing us back. Yeah.
yep. Yeah, it's kind of more in our face right now, which isn't bad. But it is a good current. All right.
This skate coming up on the right.
Oh, yeah.
Oh, yeah.
But the long nose.
There you go. I was going to ask that question. You already beat me to it.
And that was a female because I didn't see the male classlers.
I didn't
So
look, but if you didn't see him, then yeah, I would say that would be a female.
Lifted up
Turning
its
us
skirt.
into taxonomic experts here.
Yes. We passed the first quiz.
The big question is, are these male or female sponges? You got to figure that out.
Oh, man.
Oh,
Hermaphroditic. Yeah.
that was a good answer, Ben.
Do they tend to be sexually dimorphic? Is that
Not
very,
that I know of.
yeah.
That would be really interesting, though, if we found something like that. We'd be famous out of the 20 people in the world who study sponges.
20 coolest
Those
people.
would be, yeah, those would be our
Yes.
golden doubloons.
Yeah, so years ago when I used to go in the Delta submersible, it's a 2-man submersible about the size of a propane tank that you see outside of people's houses, so it's pretty small. We were in this area in Southern California, and just this whole area has lots of debris. We found a briefcase, and we collected it, and we kept saying that there was jewels and all sorts of things in it. And right when we got to the surface, it opened up and everything fell out. So we never found out what was in it or
Oh,
anything. So
no.
Mm.
I'd like to
It was
believe it
like-
was riches.
it was totally,
Not
it
to
was
rub it
the
in your
crown
face
jewels.
or anything.
They were that cool. If I could have reached out of the sub and grabbed it, then that would have been good.
I'm not sure that would have been good.
Well, at the surface, it wouldn't have been as bad.
Right.
Looking right down in a tube sponge now. There's a sea cucumber on this rock to the right.
Oh yeah, sort of draped along the
Yep.
edge.
There's a sea fan. There's a gorgonian to the left, the white tall thing, but we're not going to stay to look at it.
Look at this very large heterocone or heterocone.
Either way.
Yeah. But not all of the heterocones have that morphology of the goiters. correct?
Generally, yes. You can see some of them that just have the waffle pattern. And again, that's that question is it a different
Oh,
species
okay,
or
yeah.
not?
Thanks, Tom.
And some of them are almost satellite dish shaped, so they're not all the way up in that kind of vase look. And so they really have such a varied morphology. We call them all that, but we all call them heterocones, but they're probably multi-species. And there's a Dover sole.
Yeah, I'll do. So is it at all indicated that the morphology of their like adult forms might be induced by their local environments?
There's a steering wheel.
Yeah.
Oh, yeah.
Bubble now.
Remember the junkyard analogy?
Yep. I'm sorry, I missed the question. I was looking at the wheel.
No, I was just curious if the morphologies of the adults are kind of like induced by their environment, like the flow around them. Like, are they trying to maximize things? Is there any indication?
It could well be.
Yeah.
I mean, because in certain areas you'll see a lot more tall ones. In other areas you'll see them maybe not be as vertical. They may be bent over in one direction,
Right.
because the current's pushing them that way. So they tend to just go with the flow more. But you definitely see it with sponges going up against rocks, like this yellow one in the foreground. As instead of being a nice fan shape, it hits the rocks and then changes direction, kind of.
Right.
And here's that half alive, well, even less, a third alive and 2/3 dead one.
Wow. Interesting.
Barely hanging on.
Bridge now.
Aw.
Cat.
Did you ask to be fed, Tom?
No, well, that's probably exactly what she wants, but but she's not getting fed for 3 hours. Or it's tough
Just
life.
because we're awake.
Maybe it was a cat shark. That underwater microphone's picking up a lot.
I was gonna say, but that's exactly what they sound like.
Which many of the rockfish species actually do make sound. They make hums, they make drum sounds, they do all sorts of weird things.
Yeah, they do that with their swim bladders,
Oh.
correct?
Yep,
Yeah.
that is
So
correct.
they have muscles around their swim bladder that are like the swim bladder is like a drum, and they can contract those muscles and make different sounds. That's have you heard of the fish species, a group called croakers? That's how they do that. They croak with their swim bladder.
Wow.
Yeah.
And drums do it on the East Coast. That's why they're called drums.
That's right. The croakers and drums.
There's just so much good information coming out this morning.
Yeah. So does the ROV pick up sound so that you could, because the ocean actually makes, you can hear a lot of sound being produced by the animals. Does the ROV have that capability?
We'd be drowned out in our own sound probably.
Yeah.
You guys, yeah, we don't have any passive hydrophones or anything.
That's a question
Right.
that comes up a lot in ROV work. Most of these larger vehicles are electro-hydraulic, and the electric motor and the pump is the only thing that you would hear if there was a hydrophone down there.
Yeah.
That's a big, big swim bladder.
Well, if you hear the ROV start up on deck, you know it's really not a quiet thing.
Okay. Yeah. So it wouldn't make sense to have sound receiving equipment on there because it would just be drowned out by the ROVs.
Yes, the ROV self-noise,
Right. Yeah.
the pump and the motor screaming is pretty much all you would pick up with a hydrophone
Yeah.
for miles
Because
around.
yeah. So if there were hydrophones down here, they would be picking up the ROV. They'd be saying, "Oh, we heard something. Can't figure out what it was."
The offshore network operated by Ocean Networks Canada, there are hydrophones on the seafloor, and when we do ROV ops in the area, you can hear the ROV coming a mile away.
Oh my. Yeah. Sound travels very well in water.
Thank you. Yeah, I heard there was a recent study, or a couple of years ago, about this at the Rosenstiel School at University of Miami. There's, you know, these ultra music festival or something outdoors, and they did a experiment with all these, I want to say, toadfish, and how disoriented they were just by the noise coming from land in this little like bay there.
Yeah.
I was like, really? Adds a whole nother consideration to those type of events.
Guess you have to figure out what type of music it was,
Yeah,
whether they
right,
liked
exactly.
it or not.
We'll have to play the
Yeah,
Mozart next.
they might like reggae music, but not heavy metal.
Like Miley Cyrus, please.
At least that's how I would feel. Well, and so some species also use then sound to communicate for sexual reproduction. So that's how they attract a mate. And one of the things
Yes,
that we're
the
doing at Sinks-
midshipmen. Oh, go ahead,
yeah,
Lisa.
no, you go ahead, Tom. No, tell the midshipman story. It's pretty good.
Oh, I was just going to say one of the other species that does that is the midshipman, which for years people in San Francisco who lived on houseboats in the bay would try to figure out why is there this electrical hum, and then they all figured out that no, it's the mating midshipmen that are calling at a certain time of the year.
Wow.
Yeah.
And they
Take
would
them
all
like a
they would
long,
just
long time to figure that out.
it did.
Yeah,
They didn't ask biologists. They just asked why is there an electrical hum.
right.
It's like 'cause
Yeah, they went to PG&E.
yeah, well, everybody here blames PG&E for everything, so that's the go-to.
Yeah.
I mean, if you ever dive when there's midshipmen, if you go scuba diving, it's like somebody has a boat going over your head. It's really loud. But now you can say what you were
And then-
saying about the sanctuary, Lisa.
No, I guess I want to say in the midshipmen, I'll finish with the midshipmen. It's pretty amazing when you see they're pretty small. They're not bigger than 15 centimeters in length. Right, Tom? I'm trying to I'm holding my hand out. And that they can produce that amount of noise, that volume. Pretty impressive. No, what I wanted to say about sanctuaries is we've been for the last couple of years working with the Navy on trying to characterize soundscape. So there are hydrophones out, and we're trying to better understand what is the sound environment like, and by using that information, trying to see what are the impacts from human
0 from that.
produced sound on different species. In the past, we typically would try and better understand the sound impacts on whale species, on cetaceans, but now we're expanding it to look at other species as well.
Big siphonophore.
Yeah.
Yep, sound affects a lot of things down here. So we're getting this group of grenadiers that we've been seeing over the last, I don't know, 10 minutes or so. Are these shallow species? We have like 7, 8 species off our coast, and they're the shallowest species. And they have that black dorsal fin that sticks up, and that's one of the ways you can tell them. But of course, there's none here now as I talk about them.
A black dorsal fin sticking up?
Yeah, when one comes again, we'll see. There just happened to be 2 at once, so I started talking about them, but by the time I finished, they were gone.
Right. Because I was going to say, don't they all have a black dorsal fin? I'll have to,
Great.
I'll check.
No, they don't all have a black dorsal fin.
Okay.
These are very functional diagnostics you're giving us. Black.
Yes.
Mm-hmm.
Sort of black, you know, not too dark. those are the useful
obsidian.
ways. That's right.
Still a lot of sponges, but less of the big ones.
Dead is a big kind. I've been looking for moldy Swiss cheese, and I haven't seen any yet. So
Got to check the kitchen for that.
yeah, well, if it got that dark moldy, then I don't think I want to even check the kitchen, because that's probably not going to smell very well. I'll have my wife do that. And you're doing some great flying, because I can see everything. Appreciate it.
Great, thank you.
This is a large sponge.
Sort of fan shaped.
Yep.
We're also seeing some brittle stars now compared to the previous dive.
Yep, I see some of those. So to the left is one of those Nezumia grenadiers I was talking about.
Oh, yeah.
Of course, you can't really see. Maybe there, it's sticking up, but it's hard to see.
I did not see it.
Well, you can rewind it later and watch.
Yeah. I'll come and visit you in your lab. Tom's in Santa Cruz, and I live on the Santa Cruz side of the bay, so I could come to your lab.
I won't be there because I'm not allowed to go.
That's right. So how are you doing that? Do you have it set up at your home that you're doing the analysis that way?
Well, I don't do as much analysis anymore. I hire people to do that.
Oh, well,
That's
and
where
so how are
we have
they
all
doing?
the contractors. They're all
Yes.
doing it at home remotely on laptops with monitors or desktops with monitors. Because right now everything is digital. Back in the
Yeah.
day, we used to have high 8 and mini DV tapes that you have to change out every 40 to 30 to hour long. It just took forever. There's
Okay,
everybody's
Ben,
favorite
quiz number 2. Sorry.
Bongos.
Oh, you gave the answer.
Ah,
Male
you
or
see the
female?
Calaspers? I didn't see them.
Yes.
You got a
I didn't
50-50
see them
shot.
either.
Yeah.
Yeah, so they basically all work at home, and since we have digital, we can just share it, just like this video would be digital of Nautilus, and so people can just watch it later, and you can just do it right from your house. But I also have about 15 hard drives that I took from work, so I have the data from old cruises and things like that, so that I can check on people's data or look up questionable areas, questionable species.
Right. So I guess the limitation, because this is very high, a lot of gigabytes worth of data, is the storage of the data.
Yes,
You
it
said
is.
15 hard drives. Wow.
Last year when we did the express cruise for 30 days, we had 24 terabytes of data.
Oh, wow.
Well, I guess that's what that's what we're collecting right now on these Nautilus cruises.
A lot of data.
Right, but Nautilus they're putting theirs online for us for a lot of it, so we don't have to to walk
That's
off
great.
the ship with 24 terabytes of data. Takes forever to copy that for other people.
Yeah. Oh my God.
I'm in the middle of copying one set for Liz and her group, and I think I filled up the first 10 terabyte hard drive, and once I'm done watching video today, I'll probably start another dive. They're about 4 terabytes for each dive. Yep.
Yeah, so we've come 200 meters right now.
That's great. We can stop her right here.
Let's find the blue cheese after this. I think would make Lizzie very happy.
I wish it worked like that. Up here.
Yes. We need to put our
cat.
pig sniffing.
Well, as we transit toward the next point, if we can get as low to the bottom as possible, that's going to be our best shot to see it.
Okay.
Or if we just kind of hopscotch, go to the bottom and move a little bit and go to the bottom. And
Sure. Yeah, it'll be downslope part of it, so we can try our best to get you that view.
Roger that.
So let's just go.
Here, can we look at the grenadier to the left?
Sure.
So people want
The
to
one
see
with
what
the
I'm
black
talking about.
fin?
A black dorsal.
See the black fin? Right there.
Yes. Yes, I see it. Black. Very black.
Riley, go ahead.
But you can also see it's kind of got a hump, and it's a little taller than the other ones. Might not be, and it's got a blunt nose.
Yes, it
So
definitely has more of a
All right,
higher
go ahead and come
aspect. Yeah.
Everybody else is just going, yeah, it looks like a grenadier. Okay.
Yeah, but
So,
which
and
one?
what kind of grenadier was that, Tom?
It's Nizumia is the genus. And then it gets harder to tell on the actual species. Because a lot of them, you actually have to look at the bottom of the fish to be able to tell.
But would
And they
you
don't
call it
use
the
the
snub-nosed grenadier?
Well, there's the California, there's the shoulder spot, there's all different kind of names.
Okay. So it has lots of different common names.
Do you want to go
Well, there's different species.
300?
Okay.
Yeah.
But I was referring
It'll
to
be a
that
little
one in
longer here,
particular.
but it's only
And I would, like I
Sounds
said, I'd
like
have to flip it over to
go
really look.
around this if we want.
No, this is fine. We can just head straight over to AC. 300.
Yep. And I'll wait for you to get out.
Is there a reason we're seeing so many sponges in this area and not as many coral?
300 for our next waypoint.
You know, that was a question that we had. So basically to the west of us is Piggy Bank. And we surveyed all around the bank. And the east side, which is the side that's facing us right now, was covered in sponges. But the west side and the south side were not. And they had more corals. So that's just one of those questions that we have to figure out if it's a flow pattern, if it's sedimentation, like the sponges maybe can live in a higher sedimented area than the corals, or the other way around. So we're trying to figure that out. We're trying to figure everything out.
Yeah, and the data that we're collecting on this dive will help figure that
Yes,
out.
it will. Yep.
So this is called the Poti Peninsula, and he's one of the people that is taking all this data and putting them into models.
Correct.
Yeah.
So the current information must be a little bit more elusive than the others that we can measure directly. Is that true?
Yes, for the overall area, but some people have put current meters down in certain spots so they can get that, but it's really only for that one particular area.
Right.
So people model the currents out here, and that's what they use for distribution models of coral, sponges, fishes, because there aren't meters down everywhere.
Yeah.
You can't measure. I wish there was all those instruments on the bottom that we could all look at, but yeah, there's a nice Pharea colony over to the left.
Hopefully all those tilt loggers match the models.
They do when they add the data into the model.
Marley, I do a quick zoom.
That's fair.
That's great.
What's that behind us?
There's a Dover sole on the bottom and a Aurora rockfish above that. With a very white head.
Oh, yeah. All right, Marley, go ahead and come wide, please.
Is that another grenadier, I guess? Yep.
Yep.